Hi, my name is Pankaj Arora. I am a clinical and research fellow in Department of Cardiology and Center for Human Genetic Research at Massachusetts General Hospital. I also hold an appointment as an affiliate scientist in Program of Medical and Population Genetics at Broad Institute of Harvard and MIT. Today I'm going to talk about our work that's going to appear in the Journal of Clinical Investigation. This work has identified a novel mechanism for hypertension through the use of human genetics. The collaborative work was performed under the leadership of three experienced senior investigators at Massachusetts General Hospital. Dr. Kenneth Block, who is an established molecular biologist and did some of the early experiments on atrial natriuretic peptide in 1980s. Dr. Christopher Newton Shea, who is a worldwide expert in complex rate genetics and blood pressure physiology. And Dr. Thomas Wang, who is a national leader in biomarker epidemiology and natriuretic peptide research. In the current work, we successfully exploited human genetics to definitively establish the role of natriuretic peptide system in blood pressure regulation in humans. We determined the impact of a single nucleotide polymorphism in the setting of a rigorously controlled human physiologic study, and then we further went on and elucidated the causal mechanism by which the variant alters ANP gene expression. Atrial natriuretic peptide, as we know, is encoded by the NPPA gene. It is expressed predominantly in the heart, more in the atria than in the ventricles. The function of the atrial natriuretic peptide is to induce natriuresis through the kidneys and promote vasodilatation in blood vessels. The first evidence from population genetic studies about the role of ANP in blood pressure regulation in humans came from the work done by our group and was led by Chris Newton Shea and Thomas Wang, in which we reported the minor G allele of the RS5468 genetic variant in the 3' untranslated region of the NPPA is associated with increased plasma ANP levels, decreased systolic blood pressure, decreased diastolic blood pressure, and decreased odds of hypertension. As often is the case in population genetic studies, it is hard to compare the magnitude of genetic effects to environmental effects. So in the current study, we asked ourselves the question, how could we go from genetic association to physiology and molecular mechanism? We designed genotype-directed physiologic studies. We selected people based on their RS5468 genotype and manipulated an important environmental determinant of blood pressure regulation, which is salt intake. The second question that we asked was, how could we translate RS5468 genetic association to biological function? If we think of NPPA gene as a schematic as shown in the slide here, RS5468 is located in the 3' untranslated region. Since RS5468 was located in the 3' UTR region, and we now know that the microRNAs are post-transcriptional regulators which bind to the 3' UTR of the target genes, we hypothesized that RS5468 association could be mediated by interfering with the binding of a microRNA. We genotyped people for RS5468 and recruited 699 individuals of European ancestry. We had 23 people with AA genotype and 8 with AG genotype. Both groups were given either a high sodium diet, which was 4.6 grams per day for 1 week, and low sodium diet, which was 1/4 gram of sodium per day for a week in random order. After a week of standardized diets, subjects were admitted for an overnight visit at the Clinical Research Center. They were kept supine and ambulatory blood pressure monitoring was performed using a space lab system. The next morning, 2 IV catheters were placed for phlebotomy and infusion of 2 liters of saline over 2 hours. Venous blood was sampled hourly over 8 hours for serial and terminal pro-ANP measurements. This is a graph showing plasma and terminal pro-ANP levels on the y-axis and dietary exposure on the x-axis. As we can see in the figure, AG individuals had higher plasma levels of N-terminal pro-ANP on both standardized diets than did AA individuals. And we can also notice that the magnitude of RS5468 genetic effect approaches that induced by 1 week of 20-fold higher sodium intake, approximately 50%, which is a substantial effect on N-terminal pro-ANP levels. Next, we looked at plasma and terminal pro-ANP levels after acute saline challenge in both genotype groups. The left panel in the figure is on the background of a week of low salt diet and right on a high salt diet. The difference between the two genotypes persisted, and it appears AG individuals have increased ANP set point without altering the responsiveness to salt loading. After understanding the relative effect of RS-5468 on natriuretic peptide levels, we went on to test the hypothesis that the mechanism involves interference with microRNA binding. Bioinformatically, we looked at microRNA targeting NPPA gene with A allele. We found 3 such microRNAs, of which 2 were expressed in human cardiac tissues. We were only able to validate microRNA 425 in further experimental studies. We created reporter constructs with the NPPA 3' UTR major and minor allele. We transfected these DNA constructs in COS7 cells and co-transfected them with microRNA 425. As you can see in this cartoon right here, one would expect if we have the 3' UTR with the major allele, we would see inhibition of the luciferase activity and no effect with the minor allele construct. This is a graph showing luciferase activity on the y-axis. White bars represent transfection with control RNA and black bars represent microRNA 425 transfection. MicroRNA 425 reduces the activity of the major but not minor allele of the NPPA construct. If we transfect anti-microRNA 425, it would bind the endogenous microRNA 425 in COS7 cells, and we would expect to see reversal of the luciferase inhibition noted earlier with the major allele construct, or in other words, derepression with no effect in the minor allele construct as shown in this cartoon. We saw that the anti-MIR 425 increases the activity of the major allele construct with no effect on the minor allele NPPA construct as we predicted. To show that the single base pair change at RS-5468 was sufficient to account for its effect on NPPA expression, we constructed a modified microRNA 425 in which a single base in the seed sequence was changed to complement the minor G allele of RS-5468. In contrast to MIR 425, the modified MIR 425 selectively suppressed expression of the minor luciferase construct but not the major luciferase construct. To determine whether MIR 425 could modulate endogenous NPPA gene expression, we transfected this microRNA into iPS cell-derived human cardiomyocytes. As we can see in the figure in the left panel, compared with the negative control microRNA, transfection of MIR 425 into cardiomyocytes reduced the NPPA mRNA levels by 66% and secretion of N-terminal pro-ANP immunoreactivity by 56%, shown on the right panel. In summary, we started with a population genetic study to identify a DNA variant associated with NPPA levels. We studied the effect of the variant in response to salt loading and identified a novel microRNA regulating NPPA levels. These findings raise the possibility that the therapeutics based on anti-microRNA 425 could be used to treat disorders of salt overload, including hypertension and heart failure. Thank you for listening about this exciting journey from population genetic studies to clinical medicine.